Welcome to my presentation on the structural basis for active site probes targeting Staphylococcus aureus serine hydrolase virulence factors. So what I'm depicting here is now the structure I determined on an atomic scale with X-ray crystallography in gray carbon, in red oxygen, in blue nitrogen, and then there are also some sulfur atoms around. So the protein is made up of amino acids that have these 4 different atom types. And in this case, it's all about this active site pocket here. What I'm showing here in color is a hydrophobic region is red and a hydrophilic is blue. And this whole protein is folded this way to form this small gray pocket we saw, because that's where the serine hydrolase activity will take place. I can also show you this in a ribbon representation. So going from the first residue to the 250th residue, from blue to red. And we go along this string of amino acids. And if you just look at the sequence, you will never know how it folds in the 3-dimensional space. I can also show you all the atoms, but this gets really confusing. But X-ray crystallography has the power to show you every single atom. So again, these beta sheets here form the core of a serine hydrolase and to position this serine residue just right for the reaction. Nearby, we will see that it needs the partners to help with the reaction. And of course, I could show in the structure how this works. And here I determined a structure with an inhibitor bound to the serine. So the serine is stuck to this inhibitor. So this enzyme will no longer perform its reaction because it's bound to this inhibitor. And why is this inhibitor bound here? Because there's a hydrophobic pocket that very nicely interacts with this inhibitor. And if we look here in the surface representation, this inhibitor fits very nicely into the pocket that's right next to the serine. And why does this pocket exist in the first place? I could also show that by solving a structure bound to a substrate. So this is a C7 hydrophobic lipid substrate. And again, this substrate fits very nicely into this hydrophobic pocket. And I could again show why it prefers this C7 chain length, because if the chain would be longer, it would clash with here. It would be short, it would not utilize all these contact points. And if we want to optimize any inhibitors or probes, we now have the 3-dimensional surrounding of our initial inhibitor, and we can utilize these residues or these residues down here to increase specificity or whatever we want to modify in our inhibitor to eventually maybe get to a very specific drug. So what are the future directions? We want to determine the biological role of all FPH A to J proteins in biofilms, and then maybe identify the targets we really want to go after in a specific inhibitor and probe design. And the structure-function relationship, which is my specialty, is really what enables us to do the optimization of the inhibitors and the probes. And these inhibitors and probes are hopefully ending up then being new imaging agents, diagnostic probes, and inhibitors that ultimately will turn into drugs against Staphylococcus aureus. And lastly, I want to show you an example about FphB, where we have a specific probe already. So FphB was labeled down here with a probe that's fluorescent in magenta. And we can compare that with Staphylococcus aureus that has GFP expressed. So normally bacteria would not glow green, but GFP was added, so the whole bacteria will glow green, and FphB specific will grow in magenta. And when we overlay that, there are some differences here. So that's why we think FphB acts on the cell wall, maybe facilitate interactions or communication between bacteria. And here we can see this again over here. So this is a way of how we can use structures to design and optimize inhibitors and probes, and then using methods like this, we can show where their modes of actions is, what are their biological roles, and ultimately we want to use these tools, as I said, to target Staphylococcus aureus, because this is a serious pathogen and it will only get probably worse with strains that are resistant to our traditional antibiotics. So lastly, I would like to thank you for your attention.